Please be seated.
Hereby, I open this academic ceremony in which Ms. Claudia del Toro Runzer will defend her academic thesis Code for Bone, exploring mRNA-based strategies for complex fracture healing. I welcome you and wish you lots of success, together with your paronyms, of course. And I also welcome your supervisors, Professor van Griensven and Professor Baumeyer, and of course the audience here present and online, and obviously the members of the opposition whom you will meet later on. But before we start the opposition, we give you the opportunity to tell us what you have presented in your manuscript.
Thank you, the Prorector, members of the Defense Committee, supervisors, family, and friends. I'm very happy to be here today and talk about the research I have done during my PhD. But before I start going into my main findings, I would like to start talking about an organ that allows us to move, support our entire body, protect our vital organs, work as a mineral storage, and even produces our blood cells. I'm of course talking about our bones. And they are able to to do all these functions throughout our lifetime thanks to a process called bone remodeling, where old bone is constantly being removed and new bone is constantly being formed. This is a very delicate but important cycle. And if there is an issue in it, it can be very problematic. Let me give you an example. Imagine that you're riding your bike and that you have an accident, which results in a bone fracture. In a healthy scenario, with proper care, you will normally heal within 8 to 10 weeks. However, if there is an unhealthy scenario where you have some issue with your bone remodeling cycle, it can be that it never heals, which leads to a non-union. Some risk factors that can lead to this are some bone disorders such as osteoporosis, some genetic predispositions, your age and gender, if you suffer from an infection, if you take some specific medications, and some other disorders such as diabetes. The standard way of treating these non-unions are with autologous bone grafts, where the surgeon takes bone from one part of your body, typically your hips, and they place it into the fracture site. This works very well, however, it has some problems, because then you have two surgery sites, both with pain, and also there is a limited amount of bone that it can be extracted from there. Another option is using allografts or xenografts, where you use bone from other people or from animals. This is also a good option, however, you have the risk of disease transmission and immune rejection. An alternative is using synthetic bone substitutes. For example, the ceramics, bioactive glasses, and calcium phosphate cements. These are considered more bio-inert, but they have poor mechanical strength, so they are not very adequate for large bone fractures. Also, they are not really well integrated into your tissue, which leads us to our last category, that are the biologics. Just as the name suggests, these are molecules derived from a biological source, such as our proteins, our DNA, our cells, that we can use to treat a fracture. In our chapter number 2, we have a review article that precisely discusses in depth each one of these categories, talking about their properties, limitations, and advantages, and also future prospects for bone healing. In this thesis, however, we are going to talk specifically about messenger RNA. But what is messenger RNA? So in our body, we have cells that are constantly producing all types of protein to make everything work smoothly. These instructions for these proteins are stored in our DNA, which are always in the nucleus. However, and we use our messenger RNA to make a copy of these set of instructions to then produce a protein. So in order to use this as a therapy, scientists carefully select and design a messenger RNA and give it to cells in a process called transfection. However, as I mentioned before, DNA and proteins could also be used as therapeutic agents. The advantage of using messenger RNA is that it's considered much more affordable. Compared to proteins, you need lower doses, which reduces the risk of side effects. And also compared to DNA, it's considered safer since it doesn't integrate into the genome, so therefore there are no mutations. However, it's still not perfect because messenger RNA is considered a very unstable molecule that is also immunogenic. So in order to have an efficient messenger RNA therapy, we need two key aspects. The first one are chemical modifications that we do to the messenger RNA sequence, where we add, remove, or substitute some parts of it to protect it from degradation, enhance its protein synthesis, and also avoid immune detection. The second very important aspect are the delivery particles, where we encapsulate the messenger RNA, and this helps it to protect it from degradation, but most importantly, this allows the messenger RNA to enter the cells. Without it, it is very unlikely that the messenger RNA alone will enter the cell, and therefore there will not be protein produced. In our chapter number 3, we also have a review article where we discuss this in depth, and we also talk about different delivery routes that we can use to deliver our messenger RNA into a bone fracture. So now that we understand what is messenger RNA therapy, the question is which proteins should our messenger RNA encode? Because of course there are many proteins related to the bone healing process. However, in this thesis we focus on more morphogenetic proteins, in short BMPs, specifically BMP2 and mostly BMP7. These are very potent osteoinductive molecules that are also clinically validated. Also, in some of other chapters, we use messenger RNA coding for reporter proteins. These, for example, Metrida luciferase, are proteins extracted from this aquatic animal and have nothing to do with the bone healing process. However, they produce light, which allow us to experimentally optimize many parameters and see if a transfection works properly. In fact, we used this reported protein in our chapters number 4 and 5, where we investigated how this messenger RNA enters the cells, how it moves inside the cell, and how much protein is produced. So in order to study this, we study different endocytic pathways. You can see this as different doors that the cell has, so external material can enter the cell. And we also use inhibitors that block each one of these doors, along with a fluorescently labeled messenger RNA that is basically produces that our messenger RNA shines, so it's easy to quantify uptake using flow cytometry. What we observe with these results is that the messenger RNA complexes uses different doors in order to access the cells. However, when we see protein production, it is one door that is called caveoli-mediated endocytosis that allows the entering of particles that are of medium size compared to the other two doors that really affects or influences protein production. We further validated this with a microscope when we observed some caveoli-like structures adjacent to complexes that were being internalized. Also, we collocated around 12% of our complexes to compartments that are called lysosomes. Here, our complexes are actually being degraded, so they are not really producing any protein. And in this study, we also compared messenger RNA transfections with DNA transfections. And here we observe that in slow dividing cells such as osteoblasts, it is the messenger RNA transfections that actually produce protein levels compared to the DNA transfections. Also, we generally observe that transfections of cells seeded on a 3D scaffold produce more protein than when they are seeded in the standard monolayer culture. Moving on to our next chapter, number 6, we then we screen different lipid vectors and we see how efficiently they deliver our messenger RNA and how they produce or how they trigger osteogenesis. So we tested 6 different lipid vectors, first coding for our reported protein. And we tested different concentrations of our RNA as well as different mRNA to vector ratios for a total of 462 conditions. This is just an example of how our results look like, where the darker blue boxes are conditions where there are more protein being produced. We coupled these results with some cytotoxicity assays. Here we see that some conditions in red are where the cells have more cytotoxicity, whereas the green boxes is where the cells are more viable. So with these two data sets, we narrowed down the total number of conditions to 9, which then we use to transfect cells, but now with our BMP7 coding protein. And here is where we selected our 2 best performing vectors that were NR37 and lipofectamine messenger max. So then we tested the functionality of these assays in inducing sort of bone or mineralization matrix. So first we test some early markers of osteogenesis, which is the alkaline phosphatase staining. And we also see the presence of mineralization indicated by our alizarin red staining, and also the presence of osteopontin staining, which is a protein present in the mineralized bone. Then in our chapter number 7, we look at 3 different doses of this BMP7 RNA that is integrated into a fibrin scaffold, which also contains our human cells. This scaffold is then implanted subcutaneously in the back of a nude mice. So the very first thing we analyze here is that our messenger RNA is not spreading to other organs such as the lungs, spleen, kidney, or liver. Then we also validated that our BMP7 protein is actually being produced. At later time points, at weeks 3, 5, and 7, we also studied the kinetics of bone formation. And what we observed here was that our highest dose was actually producing significantly larger bone volumes compared to the control and the other 2 lower doses. Apart from checking mineralization, we also observed the presence of vascularization and innervation in the implant. Finally, in my chapter number 8, we studied the synergistic effect of now 2 messenger RNA molecules, BMP2 and BMP7. We started by looking this in vitro in cells cultured in a standard monolayer way, or again using now a collagen scaffold. What we observed was that using a collagen scaffold extended the duration of protein synthesis to a period of 6 to 7 days compared to the 2 days production when the cells were seeded in the monolayer culture. We also observed that the combination group using BMP2 and BMP7 significantly increased ALP activity and alizarin red staining. And also we also observed some gene expression patterns that were specific to each one of these molecules when applied individually or in combination. Then we also studied this in the same mouse model we used as in the previous chapter, where we wanted to see if the RNA was spreading to other organs, as well as if our BMP2 and BMP7 proteins were being produced. Here, when we check the kinetics of bone formation, we very drastically see that only the scaffolds that contain BMP were actually producing some bone. And interestingly, the combination group was also producing the highest bone volumes. Now, to conclude, in this thesis, we evaluated how messenger RNA entered the cells and how this drives protein production, identifying the main endocytic pathways that increase transfection efficiency. We also did a systematic lipid vector screening and also identified different material-based delivery scaffolds. And we showed that BMP7 works as a potent osteoinductive molecule, which can be further enhanced by using complementary growth factors such as BMP2 messenger RNA. And although there is a lot of work to go before we go to clinical essays, I think this study allows us to see the versatility and the potential of messenger RNA therapy that is currently able to address some challenges that probably are not being addressed by current clinical options. And before I finish, I would like to really thank my supervisors for all their support during all this time, and also my collaborators in Merlin and outside Merlin, especially the cmRNA Bone Consortium project. And of course, also my family and friends. Thank you very much. And with this, I finish my presentation, and I would like to give my word back to the prorector.
Thank you very much, dear candidate, for a very interesting presentation. We will now start the opposition. The first opponent is also the chair of the assessment committee, that's Professor Welting. He's professor in experimental orthopedics, in particular molecular biology of cartilage, in the Department of Orthopedic Surgery in the Online. Professor Welting, the floor is yours.
Thank you, Pro-Rector, dear candidate. First of all, my apologies for not being able to be actually at the location because I had some other obligations. And first of all, also, of course, congratulations with the finishing of your manuscript, which is beautifully illustrated, very carefully composed, elaborate, and I think a very sound piece of work. And of course, also congratulations to the promotion team. It's really beautiful work. So I read your thesis, and I have a couple of items that I would like to discuss with you. And let's start maybe with chapter 4. So in chapter 4, you have been investigating the transfection efficiency of plasmid DNA versus the cmRNA in 3 different primary cell types. So osteoblasts, mesenchymal stem cells, and dermal fibroblasts. And what actually I found quite interesting is that, and this is also one of your propositions, is that it seems that the cmRNA seems to be less toxic than the plasmid DNA. I found actually quite intriguing. Do you have any idea why this is?
Okay, thank you for your kind words, highly esteemed opponent. Yes, I think I have some ideas. So when we design the messenger RNA sequence, we include some modifications that are specifically designed for it to become less immunogenic and therefore trigger less immune reactions. Specifically, we substitute uridine and cytidine bases with 5-thiouridine and 5-methyl-cytidine, and these are well known to trigger to some extent some immune reaction, and therefore I believe these modifications really help the messenger RNA therapy or the messenger RNA to be less cytotoxic to some extent.
Okay, because I always thought that these modifications were more connected actually to stability of your messenger RNA. I was also thinking that the plasmid DNA somehow could have been, I don't know, how. So do you know how the plasmid DNA is being produced actually? Because I saw you bought this actually from, what was it, plasmid factory or vector factory,
yes.
Something like that. So how is this plasmid DNA produced?
Yes, actually, it can also be that plasmid DNA is usually produced in, for example, E. coli, and therefore it has some components that, it has some components that are precisely not the native components of what we found on our human cells. So
Right.
also it can be that even though it must have undergone purification steps, like for a commercial distribution, it can be that there are some probably residues, or simply that the process itself being in an external organism that is not human could also cause this. Or even sometimes they also use CHO cells, which that are human, but still they're not. Probably it's not a very physiological process. Still modified or tricked.
Okay, thank you. So I would like to move on actually to some more content-related questions. So maybe we could have a look at chapter 6. And in chapter 6, you are actually transfecting your cmRNA encoding BMP7 in mesenchymal stem cells. And what I get from it, and you nicely illustrated actually in the introductory part of that chapter, is that the whole idea is that you transfect cells with your BMP7 cmRNA. And then these cells would produce the BMP7, for example, also in the clinical situation, to then influence the de novo bone formation, right? So my first question there is, so do you envision, so the secreted BMP7, do you envision that it is more like a paracrine effect or a more autocrine effect of the BMP7? Because I mean, the paracrine effect would be more influencing the environment and the cells in the environment, while the autocrine influence would be more influencing the cell themselves. So a short answer maybe.
Yeah, I think probably more the paracrine signaling.
Right.
I think, I think it's also influencing the cells in the environment, and then this also triggers like a domino effect where other cells are being recruited, and then this goes to binds to our receptors, activates the SMAD signaling pathway, and therefore,
Because the bridge that I would like to make then is actually then to chapter 7, where you loaded your scaffolds with your mesenchymal stem cells and your lipocomplexes with the mRNA for BMP7. But if it is a paracrine effect, then how does that work? So which cell types would be involved in the bone formation, actually, in the scaffolds that you are analyzing? So that's then probably not only the mesenchymal stem cells, I guess.
Indeed, I believe that actually it can be both in that ectopic bone formation model, where it's implanted subcutaneously. I think this paracrine effect is affecting both the cells that are implanted in the scaffold itself, but also maybe the cells that are in close contact in the subepidermal area. So it can be that there are some endothelial cells present that can be also being affected. Also some fibroblasts perhaps that are in that area that can also get the BMP7.
So related to that, when I look actually at the experimental groups that you are presenting in chapter 7, was there also a scaffold group that was not seeded with the mesenchymal stem cells and only with the cmRNA BMP7?
No, actually, we don't have that control group. We only have
because that would have answered actually that question, right?
Yeah, we only have the control group with the scaffolds and cells, and the scaffolds with cells and non-coding messenger RNA. But however, it would have been also interesting probably to also investigate that condition, indeed.
Yeah, okay. Okay, I think I have 3 minutes left, if I'm not mistaken. So I would like to actually ask one of your paranymphs to read proposition number 6, if you please.
Messenger RNA therapeutics should not be viewed as competitors to other orthobiologics, but rather as complementary tools tailored to patient-specific needs.
Thank you. So the question that I would have here is, what do you regard as complementary? So what is the complementarity that you're seeking? So complementary to what, actually?
Yes. I think often in the literature, I think it's mentioned very different cases of non-unions, where, for example, if a patient has osteoporosis, or maybe if a patient has a disease such as osteogenesis imperfecta, where there is like an activated osteoclastic activity, for instance, perhaps when these patients suffer a fracture, it can be very well benefited from some other biologics, for example, microRNAs, for instance, or some proteins that, or small molecules that perhaps will inhibit this, the osteoclastogenic activity of this. This is just like an example, but yeah, it can be that we can combine perhaps this messenger RNA therapy with very severe cases with maybe cells or other monoclonal antibodies.
Yeah. So you regard it as an add-on, right? And related to that, and then I'm almost finished, because if you talk about non-unions, so these usually are large fractures. And you also discussed actually the potential benefit of BMP7 being more chondrogenic. Did you have any chance actually to assess your BMP7 messenger RNA on chondrogenesis? And that is my final question.
Yeah.
Short answer, I think.
Okay. So perhaps this ectopic model is not the best model to assess chondrogenesis. We did have some, for example, mobile pentachrome staining, which in principle can show chondrogenic, positive for glycosaminoglycans. In one of these models with the collagen scaffold, we see a bit of this happening. But perhaps in order to have a full picture of chondrogenesis, it will be better in an orthotopic model.
Yeah. Okay. Well, thank you.
Thank you.
Thank you, Professor Welting, for your opposition. Now we come to the following opponent, who's also a member of the assessment committee. That's Professor Stoddard. He's Professor in Experimental Orthopedics in the University of Freiburg. And thank you for coming all the way from Freiburg.
Dear candidate, firstly, congratulations to you and your supervisory team on a very impressive volume of work. Really enjoyed to read it. Your family, hopefully, they should be very proud. I would like to focus all the questions I'm going to give you all on the idea of how can we translate this kind of technology to the clinic. So in chapter 4 and 5, you highlight differences you see, for example, different cell types reacted to different lipids in different ways. Does that mean we're going to need to target different delivery vectors for different tissues?
Hi, Listima Poning. Thank you very much for your nice words and for your question. I believe that, since here we are focusing on bone fractures, perhaps it would be more practical to have one solution that could be probably robust enough to tackle the majority of the cases where non-unions should be addressed. But in the chapters 4 and 5, we talk about these different cells. Perhaps yes, because then it's very, we have cells that are largely post-mitotic, which are not dividing very fastly, which can really benefit from messenger RNA therapy, which doesn't rely on it entering the nucleus. Then we have some fibroblast cells that probably messenger RNA, but also DNA works good. They are really dividing very fast. So like if we generally speak about regenerative therapies overall, because maybe this can be seen regarded as like a more overbroad chapter, perhaps yes, perhaps there are different things that you can consider, not only the RNA, but maybe also the lipid vector, where you can also tune the properties to maybe have a more targeted delivery. And then also we have the scaffold part, where you can also tune many of the properties of the scaffold to achieve a very specific goal.
Thank you. And you mentioned in your answer that you have the difference between slow and fast dividing cells.
Yes.
In a healing environment, do you think you're going to have more slow or more fast, or would that affect your choice of vector?
Yes. So in a healing environment, I think, well, there are many cells being present there. We have at the beginning, we have a lot of inflammatory cells, which probably are very actively dividing. We have also human mesenchymal stroma cells in that area, which are, as we see that in there compared to the other cells, have like more like medium dividing rate. Then we probably have also some osteoblasts there, which are very slowly dividing. So I think in the healing environment, we have almost every possible scenario there, because it's a complex tissue, so you have different.
I agree entirely. And based on, because you also now have a lot of screening experience, screening these molecules, and that's often done in 2D in the lab, versus what we want to see is what's going to happen in a patient effectively. How much reliance can we have on our in vitro models, and what could we do better later?
Yes. Yeah, this is true that in vitro models, unfortunately, are not able to recapitulate everything that happens. It's impossible. But I think to some extent, they give us a good idea on if a vector is being or a lipid is being cytotoxic or not. And then it gives us an idea on which conditions we should focus and which ones we absolutely discard. But we can absolutely improve this. And one way of doing that, for example, could be using some, for example, maybe organoids or bioreactors that probably have that, where you can put some external stimuli, like mechanical properties, for instance, that can probably simulate better what is happening in a bone, where you can probably see different cells at the same time, for instance, where you can have more like a network, or like maybe some aggregates with different cells present. So I think, yeah, there are many options where we can improve and have some model that maybe can recapitulate slightly better what's happening in vitro.
Thank you. And then in chapter 8, you showed very nicely that BMP2 combined with BMP7 leads to the best outcomes, and the temporal aspect was lost, which is also a benefit for a therapeutic. So does that mean going forward, because you know these kind of projects always go forward, should we not be doing anything with only BMP2 or only BMP7? We should only be working with 2 and 7.
That's a good question, I think. Well, in this case, I think we have the advantage that both molecules are clinically FDA approved. So in terms of practicality, I think going to the clinics, it's actually possible to generate, for example, a scaffold that is maybe off the shelf that can already contain both molecules. BMP2 and BMP7 have a bit of different mechanisms of action. For example, BMP2 triggers more the early osteogenic effect, where it stimulates the differentiation of HMSCs to osteoblasts in an early stage, whereas BMP2 is more focused on a later stage where it's more important to have a more like a bone remodeling phase and to organize the collagen fibers in an adequate manner. So probably it also depends on the fracture site. Also probably it depends, but I think having both could be an interesting option, yes.
Do you see any disadvantages to taking both? Why would we not take both when it looks so nice?
Yeah, I think it would be interesting to do some orthotopic models, again, combining, for example, adding them individually or in combination. And I think it would also be interesting to do not only long bone fracture models, but also, for example, a fracture in the skull, but it has a different, it's like a more intramembranous mechanism. And in this way, we can see if one or the other or the combination works better for different locations, for example, or different fracture sizes or fracture places. Mm-hmm.
And if you had to guess, would you think you would see a difference?
Yeah, I think so. I think what we observe overall with the combination treatment is that it's like an earlier response, and also I am assuming that when we see an orthotopic model, we will probably have a bone healing that is more like nicely bridging the gap compared to probably if it's one or the other, or that is, for example, if it's BMP2, that it can be like a very robust healing, but maybe a bit more disorganized. Maybe I have hypothesizing this, but
that's what I asked you to do. So thank you very much. I hand back over to the director.
Thank you very much for your opposition, Professor Stoddard. Then we come to the following opponent, who's also a member of the assessment committee. That's Dr. De La Vega Amador. He's assistant professor in orthopedic surgery in the Department of Orthopedic Surgery in the Mayo Clinics in Rochester, USA. And thank you very much for coming all the way from the USA. University of Maastricht is always glad to have people from outside and abroad.
Welcome. Dear candidate, congratulations on this day and on this very nice piece of work, very hefty. I enjoyed it, reading it very much. Your promoters and your family should be very proud, and I especially like that the journals where you have published are very well regarded journals, so you should be very proud of this. I do have some questions I would like to ask you, and if one of your paradigms could please read proposition number 2.
Enhancing mRNA sequence design should primarily aim at improving mRNA stability, protein production, and reduce cytotoxicity as cellular targeting. Uptake and endosomal escape are largely determined by delivering vectors, chapter 3.
Notice that one of the things that's missing there is the big elephant in the room whenever mRNA is being discussed as a therapy, which is inflammation. You briefly touched on changing uridines and cytidines for reducing inflammation, but do you have any other ideas on how design of the mRNA molecules could actually help reduce inflammation? Because we know a highly inflammatory environment is deleterious for bone regeneration.
Highly esteemed opponent, thank you for your kind words and for your question. So if I understand correctly, the question is to some sort of triggering inflammation rather than avoiding inflammation.
How the design of your molecule, because you focused only on production, protein production, yes, and uptake,
yes,
but you didn't mention at all decreasing inflammation.
Okay. Yes. Well, nowadays we can use. I know that there are many groups that are using this artificial intelligence and machine learning to design messenger RNA sequences that are especially aiming for reduced inflammatory reactions. And I think the way they do this is, for example, with codon optimizations, where you can, well, I don't know exactly how it's the basis of the machine learning. I have no idea, but I know that by modifying the codons or by optimizing the codons in a way that you reduce or you can sort of choose which secondary structures of the messenger RNA you are choosing, which reduces the free energy. Then you can select the some sequences, like you can screen maybe thousands of sequences sort of all in the silico, like in your computer, and select the ones that have the rest free energy, which will mean that it will have a conformational structure or second structure of the RNA, which can really decrease the messenger RNA cytotoxicity or like immune activation. This is one way. Another way I think probably is when you use like there are other groups that have also really play along with the polyethyls, using like even branch polyethyls or even with a 5' cap, which can also to some extent influence the immune activation. So there are like some options there, but I think one of the most important overall basis or principles are really with the base modifications that I mentioned earlier.
Okay. Thank you. In chapter 6, you screened a lot of cells with a lot of vectors, Hector 93 cells and human MFEs. And Professor Stoddard already touched about how in vitro models don't usually translate about in vivo because in a fracture setting, we actually have dozens of cell types. So you briefly touched on organize. Which other cell types would you like to actually add in an experimental setting to make this a much more robust translation?
I would love to have the opportunity of combining, for example, in an in vitro setting, immune cells. Like recently, we found in our group, and this data is still not published yet, but that one of the cells that are more receptive to the messenger RNA are the granulocytes. So it would be super interesting to combine, for example, using a transfer system or maybe different scaffolds materials, ones with one type of cells like the human mesenchymal stroma cells, and another one with the immune cells like granulocytes, to see how upon transfection these cells interact, how much cytotoxicity is being produced, if this actually improves protein production, and of course also to see some functional assays where we see mineralization, if it's being enhanced, reduced, like I think these two would be very interesting. But there are many. For example, you can also test some endothelial cells to see if the presence of, for example, the BMP7 could also sort of allow some vascularization in vitro, or there are like many combinations that you can test.
And do you have any ideas on maybe targeting delivery of these cmRNA molecules to specific cells and avoid others?
So for bone-specific purposes, I think, so we are always aiming to incorporate this messenger RNA in a scaffold that will be locally placed in the fracture site, and therefore to a great extent contain the RNA in that area. Of course, there are some strategies that we can use to further improve the targeting. I was reading some groups, they work with biphosphonate ligands linked to the PEG group of the lipid vectors. And because biphosphonates really attach to the bone, to the, I think, hydroxyapatite of bone, then you can really target bone, and you can probably guarantee even more that it will not leave that bone environment. But perhaps, at least we have seen, I think, is that with the scaffold, it probably is a good level of containment, enough for it not to spread or to avoid off-target effects.
Interesting. I want to move on. Some last questions. In chapter 7, you used where you used nude mice, where you planted MSCs in the scaffold. You focused on looking at nerve ingrowth. Can you elaborate more on why you looked at it? Were you expecting nerves not to grow into it, or what was the rationale?
Yes. So, well, there are two things here. First, BMP7 is not only very, like, one of the functions that is mostly known is, of course, for bone, but it also is participating in many other systems, and it can also have some effect in bone innervation overall. I think it's also responsible for axon elongation. That is one thing. Another thing is that bone is always, like, as I mentioned before, it's not only a mineral matrix, it has blood vessels, it has nerves. So we thought that, okay, if we see already some nerves invading the scaffold area, it could be probably a positive outcome to see. That will mean that it's not only targeting our mineralization enhancement, but also innervation.
Okay, one last question, a
No,
quick
no,
answer.
I'm sorry, there's time.
Then I give the word back to her.
Thank you very much, Dr. De la Vic Amador, for your opposition. And we come to the following opponent, that's Professor Smeets. And he's professor in clinical genomics with a focus on mitochondrial diseases in the Department of Translational Genomics in the University of Maastricht. And today he has also voluntarily accepted the role of secretary of this committee, for which I am very grateful.
Thank you, Mr. Pro-Rector. My compliments for an impressive thesis, and also the layout and figures are beautiful as well. My compliments for that as well. And impressed by your thesis, I started very quickly a collaboration with your promoter. So also my compliments to the promotion team. But obviously your defense is a good opportunity to discuss your work and our plans with the real expert in the room, and that's you. So this opposition is purely self-interest. Well, what I looked at from a bit of the outside, the whole bone fracture healing is a very well orchestrated time process with all kinds of genes and proteins being active. And you choose through in two things, key drivers, and that should recover that whole orchestrated, finely tuned mechanism. Is that possible?
Highly esteemed opponent, thank you for your kind words and for your question. So I think that it is possible because the effect that BMP7 in this case triggers is like a domino effect, where even if it's one-time dose for a fracture healing, where you require like a therapy that is not like a forever treatment, but it's like for a specific window of time, I think then it's really a good option, because then you give this dose, which perhaps can be some sort of tune for a bit of more than 2 days, like maybe 7 days, using different, like, yeah.
To interrupt you, in your scheme, I see different doses and different cell types. You just give one. Is that...
different doses and different...
Yeah, I see cell types produce your BMP at different time points, different cell types produce it. So how do you tune that? Because in the beginning you need less than later on. And you need specific cell types producing it.
Yes. So I think it's possible to tune that, for example, using biomaterial engineering, for instance, where, like, in this case, we did not focus on the tuning itself so much, but I think it's possible, for example, using a biomaterial where you incorporate your RNA maybe more towards the core, for instance, and then you protect it with some extra layers on the shell sort of thing. So then you need some, you can expect that you will need some time for it in order to release, and then you will have maybe some control over the time where your cells are actually exposed to the messenger RNA. That is an option that I'm thinking.
Could be, and an alternative could be that you preload your cells, so that you have your cells ready, you preload your mRNA in it, you deliver them, and you reorchestrate your procedure in that way. But you're not such a fan of these cell type deliveries, although you show that they're very effective, I must say.
Yes, I think, yeah. So I think the thing is about the cells. In an orthotopic model, we aim to actually avoid the cells, because one of the beauties of the messenger RNA therapy is that it's very affordable, very simple in principle. So you can really go to your freezer, take out a product and implant it into the patient. If you add the cell component, then you really increase costs, time, and complexity of the entire procedure, because then you have to comply with different good manufacturing processing procedures or techniques. You will probably need some extra cost only for the isolation, then for the expansion.
But how could you reduce these costs? Because we work with, you're thinking about autologous therapy, I think, yeah. Could we use these cells off the shelf? Do you think that would be possible?
Well, there are currently different alternatives in the cell area. Some still not fully or in the process of being clinically validated, for example, using induced pluripotent stem cells, where you have some advantages. For instance, that in principle, you keep the stemness of the cells for a longer period of time, so they are more like a younger phenotype. So yeah, for example, this is a very good option that still very maybe not in the clinics yet, but it could be a potential option.
Yeah, because we're considering that in our muscle stem cell therapy as well, that you create the universal immunotolerant muscle stem cells. Would that not be something for you as well? To have that one, and you can just load you have your microRNAs or your mRNAs there, you just load them, you put them in, and you design that, and then you always have the same quality. The quality is you always have the same age. You just have to make them, and they have to be more or less immortal.
Well, if we manage to immortalize some cells and that we can verify that these cells are not going to trigger some immune rejection sometimes it's delicate when it's not your own cells, that it can generate some rejection towards other tissues. Then it can be something very interesting to look at that option, yes.
How do you would think that immortalization would happen? Do you have an idea?
I have never looked into the immortalization protocol myself, but yeah.
Yeah, for the most part, we have media which indeed immortalize these primary stem cells to almost indefinitely and at better quality than iPSC. So I think it would definitely be something which would indeed increase the efficiency and take away your argument of and you have shown that these cells can pick up your mRNA very nicely. So I think you also have the tools to explore both opportunities.
That would be an interesting option indeed, yeah.
Well, my background is genetics, so I was also surprised indeed, and we work on genetic diseases, that you say, well, you can also use your approach for genetic bone diseases, osteogenesis imperfecta. How are you going to do that? I was wondering. Yeah.
Well, I was meaning more to patients that have osteogenesis imperfecta that have a bone fracture. Okay. I have always seen it like that, not like really to address like a genetic disease only with this, because then you have much more complex scenario where you probably need to address like the entire skeleton, for instance, where that is constantly being affected by the condition.
But even if you would have these fractures, because they have fractures all the time, how would you do that? Then would you just then restore the fragile bone again, or would you then still not want to make that stronger a bit and putting some collagen 1 in it or whatever to do?
Yeah, I think what I would do in such a scenario would be, for example, of course, having our messenger RNA therapy along with some complementary therapy. I think if you inhibit sclerostin, which is, I think, activating the osteoclast, then in such cases, then at least you slow down or the very high bone resorbing activity of the osteoclast. So therefore, probably you allow your messenger RNA to work better and to actually heal the fracture site.
Even though there's a genetic defect in the structure itself.
You know,
You think more of a poor quality is better than having less of a poor quality?
Yeah, probably.
I still have time, yeah.
Short question, short answer.
Then coming back to more of a personal question, we work on muscle cells, so we also like to treat muscle injuries, but then you would like to treat them quickly. And next year we have the World Championship soccer.
Yes.
If our player, Virgil van Dijk, would break his bone in May, would you then be able to get them ready before the tournament starts? Is that also things you are working on to speed up the recovery of key players? Or do you, as you will support Mexico, don't care that you think Virgil van Dijk, get rid of him?
I think this kind of therapy is firstly addressed to people. I'm thinking that a sport person at such high level is a very healthy person. Probably that patient with a more like a standard treatment will be more than fine. However, once we see that it has such a good effect, then why not? If you can already see that it's such beneficial, you can probably reduce the healing time. So that would be interesting.
That would be definitely a commercial interest in these, also in other sports. Thank you for your answer. Thank you very much.
Thank you, Professor Smeets, for your opposition. Then we come to the following opponent, who's also a member of the assessment committee. That's Dr. Lapointe. She's associate professor in cell therapy in the Merlin Institute in the University of Maastricht. The floor is yours.
Thank you, Pro-Rector. So, dear candidate, first of all, compliments, of course, on a very impressive thesis, and also to your supervisors and to your family as well. So, of course, I do have some questions. I wanted to start actually with maybe a very general question about the design of mRNA therapies. You talk in chapter 3 about like the UTRs, cap structures, poly-A tails, how you can tune these to balance, I think you said stability, translation, and immunogenicity. And so my question is, for bone regeneration specifically, if you had to sacrifice one of these three, which would you sacrifice and why?
Yes, okay. Thank you for your kind words, esteemed opponent, and for your question also. That's a very interesting one, because all, of course, have their benefits. I think, well, actually, maybe, so in the messenger RNA sequences, you have the untranslated regions that are also parts that we have discussed in chapter number 3 that can play an important role. However, we have also seen that if you keep this to the minimal, in a minimal size, or even delete it almost, then you avoid the RNA to have some secondary structures that will inhibit the ribosomes to bind to the messenger RNA. So among all the ones that I have discussed there is actually some literature that benefits from those ones being absent. So I think that's like an easy way to say, okay, I have to get rid of one. This one is like a good option. Other than this, I probably, I think probably I have seen people also playing with different sizes of the poly-A tail and having different effects on stability of the molecule. And stability can probably also be any ways enhanced, or the RNA can be protected with a lipid vector. So perhaps if I could maybe choose another one, then I would go for that one.
Thank you. It's really interesting. I also, maybe another question about design. Earlier, you talked about the AI-driven codon optimization, which I think is very interesting. Maybe, a bit of a different type of question, but I want to know actually how you feel about that. Should we be handing over the design to AI, and maybe what are the implications of that?
Yeah, okay, that's super interesting as well. I think I have never done that myself. I have seen in some conference talks where there are people that can enhance the protein output from an RNA around like 50 to 100 times when you use some of these codon, like in silico codon optimization. So I think what they show, this is a very drastic enhancement. And I think the overall benefit of doing this is that then you can even further reduce down the doses of your messenger RNA, which we all are aiming for, to really have a very good output in the protein level, like decent amount of picograms or nanograms produced of the therapeutic RNA. So I think I think it's a very interesting technology, which probably if I would start my PhD all over again, I would probably like to explore or collaborate with someone that has access to this knowledge, and why not implement it in the work as well.
Yeah, a very clear answer. I was going to ask actually if you would kind of do this. And I mean, do you think that there's still a use for the hand design approach? Like, do you think we still need a human involved in this story?
Actually, I think both are very necessary. Even from the designing part, I think that there are only some things that you can just now that I know that you can do some simulations in silico and do this screening and see, but there is still a lot of important biological validation that needs to come out of maybe some fine tuning or with your knowledge of probably choosing which base modification, which how many percentages. So yeah, I think still like you can combine both, I think.
We still have some work to do. It's good.
Yes.
I wanted to maybe kind of follow in a bit of also some of the ethical questions around in your impact chapter, but also just now you mentioned that, you referred to these therapies as very affordable, which I think is a bold statement for any kind of advanced therapy. But so I wanted to know, do you actually see a plausible path that patients in low and middle income countries will get this therapy, or is this a therapy for the high income countries of the world?
I think the aim is that to have an effect, just as we probably observed with the COVID vaccines, that it was basically able to reach almost all parts of the world. Some of the limitations of this is that some places in third world countries probably don't have these -80 freezers that you need, or -20, depends on the technology of the RNA, that sometimes you need to store this. But I think the objective is to reach the point where it's as affordable as, of course, I know it was a bold comment to say just like this, but we do have some background there where we know how much a COVID vaccine cost to the government, to the people. So there is already some information there that indicates that it could be very affordable. And even if it's 10 times that price, or even, it's still way lower than the current alternatives of protein-based therapies, which are in around €1,000, for instance. So I think that's the main objective. I think the main objective is with this technology to really be able to reach as much people as possible.
Okay, it's good news for health equity then. Great. A question then coming from your general discussion, where you noted that the recombinant BMP7 protein was withdrawn from the market. Of course, I think a story a lot of people know. But I want to know, based on the data from your thesis, what do you think now is your strongest argument that the future of the cmRNA version is going to be different?
Yes, so I think maybe a couple of things. Perhaps the first one is that with the messenger RNA, I think one of the reasons why it was withdrawn was simply because it was not producing revenues enough. So if we have a technology that is going to be way more cheaper and the companies will be able to probably distribute it to a broader public or patients, then it already becomes probably more tempting for these companies to reopen this BMP7 area. And the second thing is that we have also seen that it's combined with BMP2, for instance, is achieving faster bone healing. So probably, companies can also consider this as well. So yeah, I think
so you see both a commercial benefit, but also a biological, clinical. Well, I hope you're right. Maybe finally, almost a question on the chapters 4 and 5. So you use the pharmacological inhibition of the different endocytotic pathways. I think it's pretty well accepted there are some off-target effects or incomplete effects also of these agents. Can you say also maybe some other approaches you could have used, or if you defend your approach and
yes,
it is complete?
Yes, it's true that it can be some off-target effects of the inhibitors. Probably one approach would be there are different molecules that can target one specific pathway. So maybe using complementary inhibitors like for the, let's say, for inhibiting clarithin-mediated endocytosis, we use chlorpromazine, but there are others. So maybe if you select others and check and compare the yield or the inhibition percentage, or yeah, like something like by using different inhibitors to see how much it actually inhibits, that maybe can be an option, like to complement your results.
Very clear, thank you. Good.
Okay. Thank you, Dr. Lapointe, for your opposition. Dear candidate, Ms. Del Toro Runzer, the time appointed for the defense of your thesis has passed, and the decree committee will now discuss the quality of your thesis and the way you defended it. And I request, therefore, that you and your company now leave the room, and after our deliberation, the beadle will come and collect you to return to this room. With that, I suspend the meeting.
All right, please. Please be seated.
I reopen this academic ceremony. Ms. Claudia del Toro Runzer, the degree committee here present has assessed the quality of your thesis and the way you defended it. And in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. And your supervisor, Professor van Grinsven, is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor, Professor van Grinsven, to now take the floor.
All rise, please.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Claudia Beatriz del Toro Runzer, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, I now present you with a degree certificate sent by the rector, the secretary, and the other members of the committee and affixed with the official seal of the university.
Please be seated.
Estimada doctora del Toro Runzer, I'm very proud to be the first one to address you like this. 6 years and 6 days ago, on December 5th, 2019, at 2:30 p.m., you came to an interview with Elizabeth and myself in the 8P room. We had just started September 1st, 2019 in Merlin, but we had luckily an EU project with 2 PhD positions. At that time, you had done an internship in Merlin with Sami in Arts Group, and you were recommended to us. This turned out to be the perfect recommendation. You came well prepared to the interview, and we have never regretted to take you on the journey of mRNA in bone regeneration. Then you started January 1st, 2020, and got acquainted very fast with the project. But as we all know, although it seems ages ago, right? 3 months later, the Netherlands went in lockdown. Pamela, Lorenzo, and I sat together, and we were able to come up with a strict plan so that people could continue working in 3 shifts in the lab. Which shift did you take?
The last one.
The last one, exactly. Always the late night shift. Even after normality returned, you were not a morning person. Despite the difficulties during that time, you had a Formula 1 start with our experiments. Elizabeth will come back to that later in her laudatio. I also read in your acknowledgement that even Lorenzo was helping you in the lab, so that's nice. I will incorporate him also in other works that we do. In our weekly meetings, time was too short to look at all your results from the previous week. So much work you did every week. At the end of your PowerPoint with the results was always a list of questions to be answered by us, so that you could continue with the same speed. We really needed to keep up with your tempo. What wondered me was that you were never into participating in conferences that much. You did participate in the MBTE in Lunteren and won there the well-deserved Best Presentation Award. Your only big conference was actually after you finished at Merlin, and this was in Boston, namely in the RNA conference. You wanted to go there mainly to make contacts with people in the field from the USA, as your now husband, Chives, was going there. You did visit your family in Mexico a couple of times during your PhD trajectory, and when you came back, you brought always some nice Mexican sweeties, pulparendo, which I loved, yeah? And I couldn't stop eating them, yeah? I think in 3 days they were gone. Whereas for Elizabeth, they were too spicy. Well, my luck, right? Because of COVID, you got an 8-month so-called NPO extension until the 31st of August 2024. You actually wanted to have finished everything by then. However, your father fell ill and you went home to Mexico to be with him and your family. Y soy muy feliz de verla a usted y la familia hoy en esta sala para la celebración de promoción de su hija Claudia. Ustedes pueden ser muy orgullosos de ella y su gran logro. Estoy seguro que su vida académica será exitosa. But then you got an offer for a postdoc position in Stanford University. However, they only give you a contract when you show them the certificate you just received. I hope they are happy with this big one. Therefore, you started writing like crazy and kept sending us drafts of papers and papers and papers. You see the result there. You had also a written review that you sent to me. It was over 100 pages, more than 500 references. I read it. The contents were actually very nice. But I knew no journal would take that. So the three of us decided together to chop it in two. And we would try somehow. I was still not very sure. But last month, we received the fantastic news that they were both accepted in highly valued journals in the field, Materials Today and Bone Research. One last paper will be submitted soon, and that's the perfect roundup of your PhD work. Otra vez, mis felicitaciones y te deseo éxito en tu viaje profesional y personal.
Gracias. Thank you.
Well, it's difficult not to get emotional after such a long dance. I am already a very emotional person, as you know me well, Claudia. But I'm going to do my best. Today is the great day for academia and RNA research. For academia, today is a day of celebration. We are marking the achievement of a new doctoral degree, yours, Claudia, Dr. Toro Runzer. And in your case, we are celebrating a remarkably hard-working person, someone who never spared an effort, a sacrifice to go beyond the state of the art, beyond any planned boundaries, to obtain the results that shape each chapter of this thesis. From the day I met you, I realized that you were highly motivated and exceptionally driven student. I have always been impressed by your willingness to work more, harder, and to push further to achieve increasingly results. Most doctoral candidates follow in normal a predefined project proposal when shaping their thesis. In your case, you went far beyond. You designed your own experiments to test a hypothesis. You immersed yourself in the literature, presented the proposals to me and Martijn, and you immediately moved to the lab to generate a wonderful data set, accompanied, as always in your work, by the most impressive and advanced microscopy images. Those data form chapter 4 of your thesis and your first manuscript. And I have learned today we will have done IE if we give you the chance to go and design your next chapter of this thesis. It is truly rewarding to see how you have developed over the years with us. You have grown into a highly knowledgeable and professional researcher and a team player, always willing to support your colleagues and transfer your knowledge whenever needed. Even after leaving our group, you dedicated time and effort to meet with Gabi, our newly arrived PhD student, and to share your expertise and pass your passion about RNA research. I thank you very much for that. Without a doubt, today we are awarding a doctoral degree to someone who has worked, studied, and prepared extraordinarily hard for it. You very much deserve this degree that you have earned. Today is also an important day for RNA research. Your thesis includes several discoveries and innovation in RNA therapeutics and propose the application in regenerative medicine, particularly for bone regeneration. You extended some of your experiments to other cell types such as fibroblasts by your own choice, opening new avenues for future research within our group. We published new and optimized transfection methodologies. For the first time, we reported mineralization mechanisms in various primary cells, and together with Martijn and Nadia, you conducted extensive in vivo surgeries to demonstrate the osteogenesis capacity of a molecule never previously studied in this context, cmRNA encoding BMP7. On behalf of Martijn and myself, we would like to express our sincere gratitude to several colleagues and collaborators who played an essential role in your thesis, Claudia. To Nadia, thank you for your outstanding and unconditional support during the in vivo experiments of Claudia. To the mRNA Board Consortium, particularly to Professor Christian Planck and the ETRIS team, and to Dr. Bobin and OSET Biosciences team for providing key material. To Nuncia and Alexander for going above and beyond to stain human nuclei in response to a reviewer request. To Paula for performing magic on a Western blot against the clock to satisfy the same reviewer. To Fergal and Johanna for a wonderful collaboration that involved many travels back and forward, and to Chris for his contact and unconditional support. We also extend our deepest gratitude to all the members of this opposition committee. I will have not wanted a better committee for Claudia than these people sitting here today and making this day possible for you, Claudia. Especially to Martin and Rodo, I would like to thank very much for doing the travel and for being here in person for Claudia. To all your colleagues joining online to support you, Claudia, as well as those who will join later to celebrate. Quisiera concluir unas palabras breves para tus padres, if I may. Que estén muy orgullosos y que disfruten mucho de este día tan especial en la vida de Claudia. Sé bien que el llegar aquí no fue fácil y que han pasado tiempos muy difíciles. Sin embargo, hoy estamos celebrando el esfuerzo de ese sacrificio de Claudia. Mis felicitaciones y mis deseos de mucha salud para ustedes y para ti, Claudia.
Thank you, Professor van Ginsveld and Professor Rosado Belmayor, for your excellent combined laudation. Dear Dr. Toro Runzer, also on behalf of the University of Maastricht, I would like to congratulate you with the degree you have acquired. I've seen a great manuscript and a great defense, and I hope you can celebrate it in the right way. And I congratulate also, of course, your supervisors, Professor van Ginsveld and Professor Rosado Belmayor, and of course your husband, your family and friends and colleagues, and well, it's a beautiful day, and congratulations. I would like to thank the members of the assessment committee for all their work, and certainly for the travel, etc., to make this all possible. And of course to the technical support team who has organized this, and the PEDEL. Without them it would not have been possible. Having said it, I'm going to close this academic ceremony. We will be staying here so that Professor Welting can congratulate you, and we are going to make a picture with Professor Welting on the background, and then we are going to make a picture at the foot of the stairs. So, but for that we ask you to leave this room. You can wait outside, and when you think when can we congratulate the young doctor, it's quite simple. You follow us. We go first, and then. Having said that, I close this meeting.